Imagine a sprawling city completely blanketed in a toxic, corrosive smog.
Right, just everywhere.
Yeah, exactly. This smog, it doesn't discriminate. It seeps into every neighborhood, every street, like every single building. But as you survey the damage, you notice this really bizarre pattern. The vast majority of the buildings, the apartments, the grocery stores, the mid-rises, they are completely fine.
Untouched.
Right. But one highly specific type of architecture, let's say, the massive high-capacity skyscrapers, they are systematically collapsing.
Yeah.
So if that toxic smog is absolutely everywhere, the immediate question isn't about the smog, right? It's about the skyscraper. Like, what is it about the internal engineering of those specific structures that makes them so uniquely vulnerable to an environment that everything else is surviving?
It's a perfect analogy, honestly, because what you're describing is one of the most stubborn mysteries in neurodegeneration today. But instead of a city, we are looking at the human spinal cord. And instead of skyscrapers, we're talking about selective vulnerability in ALS or amyotrophic lateral sclerosis.
Which brings us to our mission on this deep dive today. We are tearing into a groundbreaking 2026 study published in Cell by Gauthier and colleagues. And for you listening, we have a massive stack of data to get through today.
Oh, yeah, it's dense.
Very dense. We're talking single-cell spatial transcriptomics all the way to human postmortem chromatin mapping. We are going to unpack, step by step, how these researchers completely reframed our understanding of why certain cells die in ALS, and crucially, what those cells are actively doing right before they collapse.
So to set the baseline here for you listening, we know ALS targets motor neurons. This leads to rapid paralysis, and it's usually fatal within 3 to 5 years. Now, for decades, researchers have relied on this specific mouse model, the SOD1 G93A model.
Right, the classic model.
Exactly. This model expresses a mutant human SOD1 protein. This protein misfolds, and it creates a toxic gain of function throughout the entire body of the mouse.
And this is where that paradox comes in, right? Because that mutant SOD1 protein is ubiquitous. Like every single cell in that mouse is just bathing in this toxic protein. Yet, the disease heavily targets the alpha motor neurons, specifically the really large, fast-firing ones that innervate fast-twitch muscle fibers.
Yep, the heavy lifters.
While the gamma motor neurons right next door, the ones controlling muscle spindles, or the visceral motor neurons controlling internal organs, they're fine. They're remarkably resilient. They just keep chugging along.
Well, the metabolic burden of an alpha motor neuron is just astronomical. I mean, these cells have incredibly long axons, and they fire at intense frequencies. So that high baseline stress might explain why they're kind of living on the edge, but it doesn't explain the actual molecular sequence of their failure.
Right.
So the researchers wanted to map the internal architecture of that failure. And to do that, they deployed this massive multi-omic approach, tracking the disease temporally from early pre-symptomatic stages all the way to the end stage.
Okay, so let's pause here and do a quick bullet point recap for you listening just to lock this in. First bullet. ALS destroys motor neurons, and mutant SOD1 is a known cause. Second bullet. Mysteriously, only specific fast-firing alpha motor neurons die while others survive.
Exactly.
So I want you to think about that city covered in smog again. If only one specific type of building collapses, what is it about the architecture of that building that makes it fail? That's what these researchers are trying to map.
And the tools they use to map it are incredible. They combine 3 distinct layers of single-cell resolution.
Right, the multi-omics. Let's break those down really clearly.
Okay, so first, they use single-nucleus RNA sequencing, or snRNA-seq. Basically, this reads the active gene expression. It tells us exactly which RNA transcripts are actively being churned out by the cell at any given moment.
It's like reading the factory floor orders.
Exactly. Then they layered on SNADX-seq. Now, rather than just looking at the active RNA, ATAC-seq looks at the physical structure of the DNA itself.
Which is so cool. Explain the mechanics of that, because the spool analogy you mentioned earlier is perfect for this.
Yeah, so inside the nucleus, your DNA isn't just floating freely. It is tightly wound around these histone proteins, almost like thread on a microscopic spool.
Right.
If a gene is tightly spooled up, the cell's transcriptional machinery physically cannot reach it, can't read it. So ATAC-seq uses a specific enzyme to tag the open, unwound regions of chromatin. It gives us this literal physical map of which genetic spools the cell has actively unraveled and laid open to use.
So we have the active transcripts from the RNA seq, we have the open chromatin spools from the ATAC seq, and then they anchored all of this with MERFISH.
Right, MERFISH, which is a spatial transcriptomics tool.
Because usually you just grind up the tissue, right?
Exactly. You grind up the spinal cord, you get the data, but you lose all the physical context of where those cells actually lived. MERFISH lets us look at the intact tissue and pinpoint the exact physical coordinates of where these RNA transcripts are hanging out in the neighborhood.
Okay, so they layer these 3 massive data sets over the vulnerable alpha motor neurons. And this is where it gets really interesting. What did the temporal tracking actually reveal?
It revealed a massive shift. These vulnerable neurons, they don't just quietly succumb to the toxic protein and die. They actively execute this highly coordinated, massive molecular pivot into what the authors call a disease-associated motor neuron state.
The DM state.
The DM state, yeah.
And the timeline on this pivot is crazy because if you look at day 65 in these mice, which is early, like barely any visible symptoms, only about 4% of the alpha motor neurons have shifted into this DM state.
It's the tiny fraction.
Right. But by end stage, around day 125, it skyrockets to 35%. And they even found these pre-DMs, cells just starting to show endoplasmic reticulum stress before fully falling into the DM cluster.
The sheer scale of this genetic reprogramming is intense. We're talking thousands of genes being altered.
What exactly is the cell doing? Like, what alarms is it pulling?
When we look at what these DM cells are upregulating, it's a massive spike in stress responses. Endoplasmic reticulum stress, apoptosis pathways, autophagy, and a lot of known ALS-associated genes like SQASTM1 and TAF15.
And TAF15 is an RNA-binding protein, right?
Yeah, one that tends to aggregate in neurodegeneration. But they also heavily upregulate nerve injury and regeneration markers like ATF3 and GET43.
So they are turning all the internal fire alarms on, but at the same time, they are actively shutting down their external functions.
Completely shutting them down.
The downregulated genes are heavily enriched for synaptic transmission, axon guidance, and potassium ion transport.
That potassium transport drop is huge.
Because if you reduce potassium channel activity, you're directly dampening the electrical excitability of the cell membrane, right? The cell is literally trying to stop firing.
Exactly. It's trying to conserve metabolic energy. The entire functional identity of the cell morphs. It goes from being this highly communicative, fast-firing neuron into a silent, isolated entity that's focused entirely on internal triage and damage control.
Okay, we have a mnemonic for you listening to help remember this DM state profile. Think of the word SAD. SAD. ES is for stress pathways skyrocketing. A is for axon guidance and synapses shutting down. And D is for denervation as the neuron pulls back from the muscle. SAD.
To love that. SAD.
The cell is basically transitioning into a SAD barricaded bunker. It's locking down. So let's do our second quick bullet point recap for you. First, researchers used multi-omics to find that alpha motor neurons transition into this DM bunker state.
Yeah.
Second, they changed thousands of genes, boost stress signals, and cut off synaptic communication.
Spot on.
So here's the question for you listening. If a cell is actively changing thousands of genes to build this massive bunker, who or what is giving the orders to initiate that lockdown?
Right, because if you're orchestrating a shift involving thousands of genes, someone has to be pulling the levers. And the researchers pinpointed transcription factors as the master regulators here.
The generals of the cell.
Exactly. The master switches that bind to DNA and turn entire networks of genes on or off. They use these in silico computational models, specifically algorithms called ArchR and CellOracle, to simulate what would happen if they knocked out specific transcription factors.
So they correlated the ATAC-seq data, those open schools of DNA, with the progression of the DM state.
Yes. And the algorithms predicted which transcription factors were driving the transition. The top hits were ATF3, NFIL3, CRE3, and members of the NF12 family, which manage oxidative stress.
Okay, wait. Let's think about the therapeutic implication of this for a second. Because if ATF3 and CRE3 are the master switches, forcing the cell to shut down its synapses, stop firing, and trigger all these aggressive stress pathways, then they are driving the pathology, right?
That's what you would think.
Our immediate biological instinct would be, oh, let's design an inhibitor. Let's block ATF3 and force the cell to just keep functioning normally.
And that is the massive plot twist of this paper. The data strongly suggests the exact opposite. Those transcription factors are actually protective. Yeah. The cell isn't shutting down because those factors are toxic. It's shutting down because those factors are desperately trying to save it from the SOD1 toxicity. In fact, previous literature shows that if you artificially overexpress ALTO3 or INFIL3 in ALS models, it significantly delays disease onset.
Oh, wow. So they're the good guys.
They really are. There's even a naturally occurring human variant of CRH3 that actually protects humans against developing ALS. And the subtype specifics matter too. They found that ARNT2 and ZFH3 protect slow-firing neurons, while MECOM and LH-IX4 define fast-firing neurons, but they actually decrease during the disease, which is deleterious.
So the bunker isn't the disease, the bunker is the survival mechanism.
Exactly.
And they went beyond just algorithmic predictions here, which I think is crucial. They validated this in vitro, in a dish. They took human-induced pluripotent stem cells, iPSCs, differentiated them into motor neurons, and then artificially forced them to overexpress CRB3 and ATF3.
And the results were definitive. Overexpressing those specific transcription factors in human stem cells, even without any toxic mutant protein present, triggered the exact DM transcriptional signature.
It forced them into the bunker.
Yes. It proves that the DM state isn't just some chaotic, passive degradation of a dying cell. It is an active, highly conserved regulatory response.
Which is just a huge paradigm shift. Let's do our third recap. Bullet 1. Master switches called transcription factors like ATF3 and CRB3 drive the DM state. Bullet 2. This is an active, protective, cellular attempt to survive the toxic environment, and it was validated in human stem cells.
Perfect summary.
So the question for you listening now is this. If the motor neuron is barricading itself in a bunker and fighting for its life, what is happening to the surrounding neighborhood, like all the other cells in the spinal cord?
And this is exactly where that spatial context from the Murfish data becomes so critical. We have to look at the non-cell autonomous toxicity, because ALS isn't just about motor neurons. It is heavily driven by the surrounding environment, the glial cells.
Right. What did the spatial maps actually show?
Early in the disease timeline, long before widespread neuronal death, the microglia and macrophages, which are basically the immune cleanup crew of the central nervous system, they're already swarming. And they are specifically clustering around these vulnerable alpha motor neurons.
But how do they know? Like, if the neuron is locked inside a bunker with the blinds pulled down, how do the microglia know exactly which houses to swarm?
The spatial transcriptomics revealed the mechanism. The DM motor neurons are actively secreting molecular SOS signals. They highly upregulate these ligands signal flares, basically like CSF1, IL-34, and LEG-ELS3.
So the barricaded cell is shooting a CSF1 chemical flare out the window.
Literally. And using an analysis tool called CellChat, the researchers mapped this active CSF1 receptor signaling axis. The DM neurons are directly communicating with the CSF1 receptors on the microglia.
Okay, so it's like a burning house calling the fire department.
Exactly. The local microglia detect the flare, they rush over, and they initiate an inflammatory response.
But in a healthy brain, that immune response would just clear out debris and support the cell, right?
Normally, yes. But in the context of ALS, this chronic, intense signaling seems to push the microglia into a hyperreactive state. They overreact. They release toxic cytokines. So instead of just putting out the fire department shows up, completely panics, and starts hosing down the entire neighborhood with inflammatory chemicals.
Man, that's devastating.
It initiates a catastrophic cascade. As the disease progresses, this glial reactivity spreads everywhere. Astrocytes become reactive. And the researchers even found a rise in something called disease-associated interneurons, or DIs.
DIs, right. These are neighboring neurons that don't even control muscles directly.
Exactly. But under the onslaught of this neighborhood neuroinflammation, they start adopting the exact same stress and amyotrophic programs as the DM cells. The whole neighborhood goes toxic.
Okay, time for recap number 4. First bullet. DM cells send out chemical SOS signals like CSF1 that attract microglia. Second bullet. This localized immune response eventually triggers a widespread toxic neuroinflammation that harms the whole neighborhood.
It's a chain reaction.
So here's the big skeptical question for you listening to ponder. We've seen all this incredible data, but it's in a mouse model with a very specific, artificially high genetic mutation. Does this perfectly orchestrated mouse bunker actually translate to the messy, complex reality of human ALS patients?
Yeah, that is the ultimate translational question. Because human ALS is notoriously heterogeneous. You have C9orf72 mutations, you have TDP43 pathology, and the vast majority of cases are completely sporadic, no known genetic driver.
Right. It's incredibly messy. So does it hold up in humans?
To figure that out, they needed human post-mortem snRNA-seq data. But translating this to human tissue is a nightmare, physically speaking. alpha motor neurons have these massive cell bodies called somas. When human tissue is snap-frozen after death and then later processed to isolate the nuclei, those giant somas act like brittle glass.
They just shatter.
They absolutely shatter. The numbers in the paper highlight just how brutal this is. Out of over 770,000 nuclei sequenced in their human dataset, only 150 intact alpha motor neuron nuclei actually survive the quality control filters.
Which is nothing. You can't build a statistically significant model on 150 cells. It sounds like a total dead end.
It really did. But the researchers developed this brilliant workaround. They realized that when those massive motor neurons shatter, they don't just vanish into thin air. They leave behind cellular debris, large fragments of the neuron that still encapsulate the cell's RNA transcripts.
And they called these alpha frags.
Alpha frags, exactly. By sequencing these alpha frags, they were essentially sweeping up the shattered pieces of the transcriptomic puzzle from the lab floor and computationally piecing the motor neurons back together.
Which is just genius. It allowed them to recover tens of thousands of profiles from cells that were physically destroyed.
And analyzing those alpha frags yielded the ultimate proof. They looked at spinal cords from patients with familial SOD1, C9orf72, and sporadic ALS. Across every single clinical subtype, the human alpha motor neurons and the alpha frags exhibited the exact same DM signature they mapped in the mice.
So the massive upregulation of stress, the deep downregulation of synapses, it was strictly conserved in humans.
Completely conserved. And they even found clear signatures of TDP43 pathology in the human alpha frags, which the mouse model doesn't even have.
Unpack that a bit. What is TDP43 doing?
So TDP43 is this nuclear protein that mislocalizes in about 97% of all human ALS cases. When it leaves the nucleus, it fails to regulate RNA splicing properly. This leads to what we call cryptic splicing. Essentially, the cellular machinery makes incorrect cuts in the RNA, inserting nonsense sequences into critical genes like STMN2 and UNC13A.
And they found these exact cryptic splicing events in the human DM cells.
Yes, but here's the kicker. Despite the presence of this human-specific TDP43 pathology, the downstream bunker state was identical to the mouse.
Which implies that the DM state is this universal cellular endpoint for motor neuron stress, regardless of what the initial toxic insult was.
Exactly. But they took it one step further. They connected the physical chromatin structure to human population genetics.
Oh, right. The GWS data.
Yeah. They took the ATAC-seq data, remember the 3D map of the unwound DNA spools in the mouse DM cells.
Yeah.
They mapped those precise genetic coordinates to their equivalents in the human genome. Then, using advanced statistical modeling tools like partitioned LD score regression and HMAGMA, they cross-referenced those regions with human GWAs for ALS.
And just for context for you listening, GWA Genome-Wide Association studies these databases show us the exact single-letter DNA mutations across millions of people that statistically correlate with a higher risk of developing ALS.
Right. So they're basically asking, do the genetic mutations that cause ALS in humans happen to sit in the exact same physical locations that the cell unravels to build its bunker?
And what did they find?
The overlap was highly, highly significant. The human orthologs of the DNA regions that open up during the mouse DM state are heavily enriched for human ALS genetic risk variants.
That is mind-blowing. So the genetic architecture the cell relies on to execute this survival state is the exact same architecture where human genetic vulnerability hides.
Yes. If a patient has a mutation hidden in a gene required to build the bunker, and the cell is suddenly forced to unravel that spool to survive the toxic smog, the mutation compromises the bunker. The survival mechanism fails.
It perfectly ties the single-cell epigenetics back to human population risk. It proves the DM state is fundamentally intertwined with how humans actually get ALS.
Exactly.
Okay, our final recap, number 5. Bullet 1. The DM state is strictly conserved in human ALS patients, proven via those shattered alpha frags. Bullet 2. The chromatin regions driving this state perfectly overlap with the DNA mutations that put humans at risk for ALS.
Yep, that sums it up perfectly.
So as we wrap this deep dive up, I want to pose a question for you listening. Think about what stands out to you here. Even though humans get ALS from completely different triggers than this specific mouse model, the motor neurons all end up adopting this exact same DM survival state. It completely upends the traditional view of neurodegeneration.
It really does.
Yeah.
Historically, when we see a diseased cell changing its transcriptional profile, the therapeutic instinct has always been to view that change as the pathology itself.
Right.
The goal has been to find drugs that block the transition to force the cell back into a normal state. But currently, ALS treatments are very limited, and this paper completely changes the target.
It forces a total paradigm shift. If this DM state is an active, programmed attempt by the cell to survive, driven by master switches like CRBEB3 and ATF3, what if future therapies shouldn't be trying to stop the cell from changing? What if we should be intervening to help it complete the bunker?
Exactly. Supporting the survival mechanism instead of fighting it.
Imagine if we could design drugs to supercharge those protective transcription factors the very moment someone is diagnosed. We might be able to stabilize that bunker before the toxic microglial response burns the neighborhood down, turning a fatal collapse into a permanent, stable cellular lockdown.
It's a completely new way to approach the disease.
So I'll leave you with this final provocative thought. Next time you read an article or a headline about neurons dying in a neurodegenerative disease, ask yourself, are those cells just passively dying? Or are they desperately fighting back and we just haven't figured out how to hand them the right ammunition yet?